Hello children, this video is in continuity with the previous video of the chapter Plant Tissue. The main objectives that we will cover in this video includes anatomy of a dicot leaf or a dorsiventral leaf and anatomy of a monocot leaf or an isobylator leaf. Now let's discuss the anatomy of leaf. The leaves are of two types depending on the arrangement of the tissue. Dorsiventral leaves, which are present mostly in dicot plants. Isobylator leaves, which are present in monocot plants. Dorsiventral leaf or a dicot leaf. A dorsiventral leaf receives more sunlight on the upper surface, and this accounts for the specific distribution or arrangement of tissues within a leaf. Here, dorsi word stands for dorsal surface and ventral word stands for ventral surface. Dorsiventral leaves are the leaves which are having more number of stomata on ventral surface as compared to that of dorsal surface. Here dorsal means upper surface and ventral means lower surface. So dorsiventral leaves are the leaves which have more number of stomata on lower surface as compared to that of upper surface. Epidermis. Epidermis is the outermost layer. It is present on adaxial as well as abaxial surface. Here adaxial means upper surface and abaxial means lower surface. Epidermis present on adaxial surface is called upper epidermis. Epidermis present on abaxial surface, that is on lower surface, is called lower epidermis. Epidermis consists of a single layer of compactly arranged living cells. Intercellular spaces are absent and chloroplast is also absent. The outermost walls of epidermal cells are cutinized. Cutin is present in the form of a layer called cuticle. The cuticle present on upper epidermis is comparatively thicker than the cuticle present on lower surface. Cuticle checks the transpiration. Stomata are present on the epidermis and the number of stomata varies in case of dorsiventral leaf. Stomata controls the exchange of gases and escape of water vapor. Epidermal hair may be present or absent. Epidermis protects the inner tissue. It checks transpiration. It regulates the exchange of gases between internal atmosphere of leaf and external atmosphere. Next is mesophyll cells. It consists of several layers of parenchymatous cells which contain chloroplast. It is differentiated into following layers. First one is palisade parenchyma. Palisade parenchyma is present beneath the upper epidermis. It consists of elongated and columnar cells. You can see here in this diagram, these elongated cells are called palisade mesophyll. Palisade parenchyma is present beneath the upper epidermis. You can see here these green colored elongated cells. These represents palisade parenchyma. The cells are arranged in one or two layers at right angle to the upper epidermis. If present in two layers, the cells of outer layer are long and elongated and the cells of inner layer are short. Cells are thin walled and are closely packed. Intercellular spaces are present but they are narrow. These cells contain numerous chloroplasts which are present close to the cell wall. Palisade parenchyma carries out photosynthesis and synthesizes the food material. Next is spongy parenchyma. Spongy parenchyma is present between palisade parenchyma and lower epidermis. The cells are irregular in shape and loosely arranged. Intercellular spaces are many and are broad. The cells contain chloroplast. The color of upper surface is different from the color of lower surface as the number of chloroplast in palisade parenchyma is more than that of spongy parenchyma. Hence, the lower surface of the leaf is pale green in color and the upper surface of the leaf is dark green in color. The cells of spongy tissue carry out photosynthesis and synthesize food materials. The intercellular spaces of spongy tissue helps in aeration. After this, next is vascular bundles. Vascular bundles are distributed within the mesophyll cells. They are arranged in the form of veins. The veins give mechanical support and helps in conduction of water, mineral ions and food material. Vascular bundles present at the base of leaf lamella are broader and those are present on the apex are narrower. The vascular bundles are conjoint, collateral and closed type. Each vascular bundle shows xylem towards the upper epidermis and phloem towards the lower epidermis. Xylem and phloems are present side by side on the same radii. Hence, bundles is described as conjoint and collateral. Cambium is absent between xylem and phloem, hence the bundle is described as closed type. Xylem consists of xylem vessels, xylem tracheids, xylem fibers, and xylem parenchyma. Xylem consists of one or two tracheids and they show annular or spiral thickening. Sometimes it may consist of one tracheids. Phloem consists of narrow sieve companion cells and phloem parenchyma. Phloem may be absent in ultimate branches. Each vascular bundle is surrounded by single layer of parenchymatous cells. This layer is called as bundle sheath. The cells are compactly arranged, which means that there is no intercellular space that is present between bundle sheath cells. The cells may or may not contain chloroplast. The bundle sheath extends on both the sides of epidermis. This extension are called bundle sheath extension. The extensions are parenchymatous. They help in conduction of material between the vascular bundles and mesophyll cells. This is all about the structure of a dicot leaf. Now, let's discuss the structure of isobilateral leaf or monocot leaf. An isobilateral leaf receives almost same amount of sunlight on both the sides. Here word iso means same, bi means two, lateral means surfaces. So, an isobilateral leaf is a leaf on which stomatas are equally distributed on both the surfaces. The mesophyll is not well differentiated into palisade and spongy parenchyma. First, let's discuss the structure of isobilateral leaf. It consists of epidermis. Epidermis is the outermost layer. It is present on both adaxial as well as abaxial surface. The epidermis present on adaxial surface is called upper epidermis and the epidermis which is present on abaxial surface is called lower epidermis. Epidermis consists of single layer of cells. The cells are living, tubular in shape and compactly arranged. Chloroplast is absent. The outer wall of epidermal cells are cutinized. The cutin forms a layer called cuticle. Cuticle checks transpiration. Stomatas are present on both the epidermal layers almost equally. Beneath the stomatas are present air chambers. Stomata controls exchange of gases and escape of water vapor. Epidermal hairs may be present. Upper epidermis shows large thin-walled cells present here and there. These cells are called bulliform cells. They are highly vacuolated and are filled with water. They are hygroscopic and help in rolling off leaf lamella during unfavorable environmental conditions. Epidermis protects the inner parts. It checks transpiration and it regulates the exchange of gases and escape of water vapor. Then is mesophyll cells. Mesophyll is the ground tissue of the leaf. It is present between epidermal layers and it is not differentiated into palisade tissue and spongy tissue. It consists of compactly arranged thin-walled isodiametric cells. Intercellular spaces are present but are narrow. The cells contain numerous chloroplasts. They carry out photosynthesis and synthesize food material. Then is vascular bundles. Vascular bundles are present within the mesophyll. They are arranged in parallel series and are collateral, conjoint and closed. Xylem is present towards the upper epidermis and phloem is present towards the lower epidermis. Xylem consists of vessels, tracheids and xylem parenchyma. It helps in conduction of water and mineral ions. Phloem consists of sieve tube and companion cells and helps in conduction of food material. Each vascular bundle is surrounded by a bundle sheath. It is parenchymatous. The cells are large, thin-walled. They contain chloroplast. Bundle sheath extends on both sides of epidermis. These are called bundle sheath extensions, which are sclerenchymatous in case of isobilateral leaf but were parenchymatous in case of dorsiventral leaf. Next is difference between dorsiventral and isobilateral leaf. As we people have already discussed, dorsiventral leaves are the leaves which are present in dicots. Isobilateral leaves are the leaves which are present in monocots. Dorsiventral leaves are the leaves in which stomatas are present more in number on lower surface as compared to that of upper surface, whereas isobilateral leaves are the leaves in which stomata are almost equally distributed. In case of dicot leaf, guard cells are kidney shaped, whereas in case of isobilateral leaf, guard cells are dumbbell shaped. In dorsiventral leaves, bulliform cells are absent, but they are present in case of monocot leaf. Next, we will talk about mesophyll cells. Mesophyll is well differentiated into palisade and spongy parenchyma in a dicot leaf, but it is undifferentiated in isobilateral leaf. Dorsiventral leaves have vascular bundles which are arranged irregularly, but they are arranged in rows in case of isobilateral leaf. In dicots, bundle sheath cells are colorless, whereas in case of monocots, bundle sheath cells are chlorophyllous, means they contain chlorophyll. Bundle sheath extensions are parenchymatous in case of dorsiventral leaf, whereas in case of monocot leaf, bundle sheath extensions are sclerenchymatous. Protoxylem is almost indistinguishable in case of a dicot leaf, but it is distinguishable in case of a monocot leaf. Thank you everyone.